Yasuhara. Yasuhara is a freshman at Wednesday College. She has a strong passion for biological sciences, chemistry, and medicine. And I think for science fairs and conferences. Yasuhara is particularly interested in the connection between biology, engineering, medicine, and technology.
Thank you for your introduction. I'm going to announce Alyssa Yasuhara, who is a ninth grader attending Newton North High School. She has a strong passion for mathematics and sciences and is currently studying biomedical science. Alyssa has participated and attended various science fairs and conferences, including her Japanese school science fairs, which she has participated in for 7 consecutive years. Alyssa is particularly interested in the connection between biology and technology, and today she will be presenting her work on cryopreservation using sugars on brine shrimp.
My name is Alyssa Yasuhara, and I attend Newton North High School, and I'm a freshman there. And I'd like to thank ASSET, the community and the members, as well as the organization for giving me this opportunity to present today. And today I'll be presenting to you brine shrimp cryopreservation. Now you may ask, what is brine shrimp? Well, brine shrimp are these types of crustaceans that live in Great Salt Lakes, and they're able to withstand waters of high salinity and as well as stressful environments. And studies have found that their eggs, which are called cysts, can become dormant, which means that they can live as in low metabolic states for long periods of time, meaning that they don't need water. They are able to live without oxygen as well as light. And this has been a very miraculous and interesting study for a long time. Now, this is a video that I have filmed myself, and this is just showing the movements of baby brine shrimp, which are newly hatched brine shrimp. And as you can see in the video, they swim very rapidly and quickly. And you may see two different types. They are the same type of brine shrimp, just in different developmental stages of their lives. So the darker brown represents brine shrimp that have like newly hatched, very, very new. And the other type, which are slower moving and are more transparent than the other one, represents like them in 1 or 2 days after they have hatched, as you can see here. Now, why did I choose to study cryopreservation? So cryopreservation is used by many medical professionals, specifically for preserving reproductive cells, stem cells, as well as using them for organ transplants. And many medical professionals have used this and found solutions to basically cryopreserve small scale cells such as single cells. And they have used methods such as slow freezing and vitrification. However, it is shown that it is unsuccessful to larger scale systems such as organs, and due to like uneven cooling as well as ice formation. So these scientists and researchers can also learn from nature, as there are organisms out in nature that use successful cryopreservation techniques, and this can lead to better insight and solutions for cryopreservation. So an example in nature would be the American wood frog, which they are a type of frog that are able to completely freeze their bodies for hibernation, whereas regular frogs are not able to withstand the low temperatures that these frogs are able to. They use cryoprotectants such as glucose, which is a type of sugar, and they help them maintain their cellular structures and prevent ice crystallization. To further explain the mechanisms of this glucose, when the temperature drops during the winter, the frog's body signals the liver to exert higher concentrations of glucose. So studies have also found that there is a different type of sugar called trehalose, which is also found in American wood frogs. And these two kind of help the wood frog minimize cellular dehydration as well as destruction by stabilizing the membranes and as well as the proteins. So as you can see here, glucose is a monosaccharide molecule, and trehalose is a disaccharide, which means that there are two glucose molecules bonded together. Now, the foundation of my brine shrimp research was I was introduced to brine shrimp when my pet betta fish wanted to have some more nutritious food, because previously I was using these daily pellets, which were advertised to have nutrition. However, my betta fish fell ill, and so I wanted to turn to better alternatives of food. So I turned to live foods such as bloodworms and brine shrimp, and I found that brine shrimp were a very accessible and easy way to feed my betta fish. So these brine shrimps are actually very easy to hatch on your own, as they only need water, salt, and a little bit of oxygen. So I bought this packet of brine shrimp eggs, and I forgot about them for a couple years. And I came back to them, finding them in my pantry. And I was able to successfully hatch these brine shrimps. And it led to the question of the capabilities of brine shrimp, because if their eggs can withstand no oxygen, low temperatures, then what can these creatures really do? So I decided to research more about the brine shrimp. Now, the foundation of my experiment came from the research of brine shrimp eggs, as well as the American wood frogs. And as we saw previously, these two organisms are able to freeze themselves completely and keeping their organs and cellular structures intact. So I thought, why not try freezing live brine shrimp, as their eggs are found to be able to be frozen, and as well as using techniques of the American wood frog, why not try and figure out different cryopreservation techniques? So for my experimentation, I took these steps. First, I hatched my brine shrimp in an air pump circulation system for 36 hours. Then I created solutions of different types of sugars, maintaining appropriate concentrations of them. Then I incorporated the brine shrimp to the mixture that I created, and I set it for 1 hour to ensure the absorption of the sugars through their bodies. Then I froze these brine shrimps at -20 degrees Celsius for 24 hours. And lastly, I thawed them slowly at room temperature, calculating and counting the survival rate of the brine shrimp. Now, this is just a visual representation of my experiment. As you can see here, this is just the circulation pump system that I created using a soda bottle. And as you can see, it's very easy to hatch these brine shrimp at home. So this is what I used. And then these plates are what I use to distribute the brine shrimp with my sugar solutions, and I froze them. And unfortunately, as you can see here in the graph, none of the brine shrimp survived, meaning that this had 0% success rate. And I concluded that because -20 degrees Celsius is at such a low temperature, this may have been too stressful of an environment for the brine shrimp. So I decided to test this theory again by raising the temperature, which I experimented this at. So I changed one factor, which is the temperature again. And instead of using -20 degrees Celsius, I used 4 degrees. And I took the same steps as before and saw the progression of this. So as you can see here, I used the same materials as well as the same plate and froze this for 4 degrees for 36 hours. And the results are very surprising. As you can see here, I only used 3 sugars because I knew that these would demonstrate the most effective strategies of cryopreservation. So as you can see, glucose had actually the lowest survival rate out of the 3, and trehalose had the highest, meaning that they had the most amount of brine shrimp survived after the 4 degrees in the 36 hours. And the second successful one was the GNT, which is the combination of the two. And yes, these are the results of the experiment. And so we were able to conclude that the -20 degrees Celsius was too extreme for the brine shrimp. However, the 4 degrees was also an effective method of testing the resilience of brine shrimp. And we found, again, that trehalose had 94% success rate here compared to glucose, which only had 72% success rate, 72%. And so we can conclude that trehalose may be more prevalent in cryopreservation in nature and survival methods of certain organisms, such as the wood frog. And so we can also implement this information into medicine. And if we develop this further, we can possibly find better solutions to cryopreservation techniques. The next steps of this experiment is to further test the capabilities of the brine shrimp, because previously we only were able to incorporate the sugars through the absorption of their bodies. However, I think there is a better solution to that, and we're trying to find better insights into that. But I think that by doing this, we are able to fully utilize the cryoprotectant for the brine shrimp, allowing more extreme freezing tests and testing the resilience. And that is the end of my presentation. Thank you for listening. Yes.
Did you publish your results somewhere?
No, I haven't. No.
Question number two, did you search for any cryopreservation, any companies that are preserving organs using your technique or similar to that?
So many organ transplants don't use sugars in cryopreservation, but they use slow freezing as well. But the cryopreservants that I tested in this were not used in organ transplants, and I used examples from the American wood frog, which is a live organism that is able to fully utilize this cryopreservant. So that is a sample that I used to conduct my study.
Where did you get like the Trellos and Lugos and all those molecules?
So I did a little summer research I interned at a summer research program, and they provided me with these sugars at their lab. Yes.
That was a great talk, really enjoyed it. I'm wondering about the Trehalose percentage or concentration that ended up working for you, actually making other solutions and testing it. So I was curious what that insight was.
Okay, so for most of the sugars, I used 0.1 molars or 342.3 grams per molar, and this did vary based off of each sugar that I used. So it really depended, but I think this is around the number that I used for Trehalose and the other sugars. Yes. Thank you.